cell membrane are present in plants, algae, as well as in the case of animals also. Ribosome is the only non-membrane bound organelle that is present in eukaryotic cell, because eukaryotes is distinguished in two ways. Functions of plasma membrane: it allows passage of only certain selected materials. That is why plasma membranes are selectively permeable in nature. You don't get to find plastids in animal cells. Plastids are the cell organelles present only in the plant cell. Hello everyone, a warm welcome to the session and revision class for first PUC Biology. I'm Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session, we will learn about the cell, that is the unit of life. So we will learn about the synopsis, that is of this chapter, what and all contents are there in this chapter, and then move on to learning the important 2 marks question and answer and 5 marks question and answer. So from this chapter, 1 2 mark question will appear and 2 5 marks question will appear. So therefore, this chapter carries a total weightage of 12 marks. From this chapter, MCQs will not be asked, meaning 1 mark questions will not be asked from this particular chapter. So you can only concentrate according to the blueprint that is given by the PU board. You can only concentrate for 2 marks and 5 marks from this particular chapter. So let's take a look into what and all contents are there in this particular chapter to just brush up. So we learn entirely about the cell. So what are the components present in the cell? So we have the cell is of prokaryotic cell and eukaryotic cell, wherein the major difference between a prokaryotic cell and a eukaryotic cell is the nucleus, right? Because the prokaryotic cell will not have a true nucleus, meaning the nucleus is not covered by any nuclear membrane. Whereas in a eukaryotic cell, they will have a true nucleus, wherein the nucleus is covered by a nuclear membrane. So in prokaryotic cell, the cells are identified based on the shape and based on the organization of the cell. What all it is made up of based on that. So based on shape, say for example, bacteria. So bacteria is a prokaryote. We all know that, and they are unicellular prokaryote. So based on the shape, the bacteria can be of different type. Coccus, meaning they are round shaped. Bacillus, meaning they are rod shaped. Vibrio means they are comma shaped, and spirillum means they look like a ribbon or they look like a spring, spirillum. So that is spirillum shape. And based on the organization, that is how their cell envelope looks, that is the cell covering, what is it made up of, what are the cytoplasmic contents that are present in them, and how their surface looks based on them, or what components are present on their surface. Based on that, we'll talk about it. So first, based on the cell envelope. So cell envelope means cell covering. So what is it made up of? So the cell of a prokaryote is covered by a cell wall, and it is covered by a cell membrane, and it is made up of glycocalyx. So what is the component of the cell envelope? It contains a compound called as glycocalyx. Next, moving to the inner content, that is the fluid matrix that is present in the prokaryotic cell, which is the cytoplasm. Now inside the cytoplasm, which is a granular fluid matrix, they contain the matrix will be present, which is granular in nature, and it contains the nucleoid. That is, it contains in the nucleoid, it can contain the chromosomes and all that, and it contains the mesosomes. Then it has the ribosomes, some inclusion bodies, and some gas vacuoles. Now these inclusion bodies are nothing but the food which was stored in the form of inclusion bodies. So they are in the form of synophysin granules, then they can be in the form of phosphate granules, and they can be in the form of glycogen granules. So food can be stored in the form of inclusion bodies, in the form of synophysin granules, phosphate granules, or glycogen granules. Next, talking about the surface. So these bacteria, they tend to move. So for locomotion, like the eukaryotic organisms, they do not have special structures for locomotion. So how do they move exactly? So on their surface, they have structures for movement, which is nothing but called as flagella, and they have pili, and they also have finger-like projections called as fimbriae. So this pili, apart from helping the organism, so this pili that is there, it is made up of, they are tube-like structures made up of proteins, and they also help in the movement of the prokaryotic organism. And flagella, they are whip-like structures which also helps in the locomotion of the prokaryotic organism. Whereas fimbriae, they are special structures which are present in some of the bacterial cells that live in water, that helps those bacteria, say for example the cyanobacteria that lives in fresh water, they help these bacteria to attach to any substratum such as rocks, wood, or anything that is there in the water. So for that, fimbriae is helpful. So these are the surface structures that are present in the prokaryotic cell. Next, moving to eukaryotic cells. So eukaryotic cells, they also have a cell wall, cell membrane, and the protoplasm. Now, cell wall. So eukaryotes, it is nothing but, here I have divided it into the algal cell wall and the plant cell wall, because there is a difference, because the composition of the algal cell wall and the plant cell wall is different. Now, what is the cell wall of algae made up of? It is made up of cellulose, it is made up of galactans, mannans, and it is made up of calcium carbonate. Plant cell wall, it is made up of cellulose, hemicellulose, pectins, and proteins are present in the plant cell wall. Next, moving to cell membrane. So cell membrane, usually it is a bilayer, that is, and cell wall is present only in plants. In eukaryotic organisms, cell wall is present only in plants, that is in algae as well as plants. But in animals, in eukaryotes, that is the animals, the cell wall is absent. So in plants and algae, the cell wall is present, whereas the cell wall is absent in animals. So you have to remember this. But cell membrane are present in plants, algae, as well as in the case of animals also. Now, what is the cell membrane made up of? The main composition of the cell membrane is phospholipids. It is made up of phospholipids, and these phospholipids are arranged in two layers, wherein they have their hydrophobic tail towards the center and hydrophilic head towards the outside. And they are made up of proteins, and they are made up of also oligosaccharides. Now, in proteins, they contain two types of proteins. One is called as the integral proteins, and the other one is the extrinsic protein. Integral means they are embedded within the lipid bilayer. They also act as carrier proteins, and the extrinsic proteins are the ones which are present on the surface of the lipid bilayer. And talking about the protoplasm. So protoplasm is nothing but both the nucleus along with the cytoplasm, we call it as a protoplasm. So in the protoplasm, they have the granular matrix which is called as the cytoplasm, and also nucleus is present in them. So this is about the eukaryotic cell. Now in the cytoplasm, what all components are present, or what all cell organelles are also present, we shall look into. So cytoplasm consists of the matrix, then it has double membrane bound organelles, and it also has single membrane bound organelles, and it has non-membrane bound organelles. So ribosome is the only non-membrane bound organelle that is present in eukaryotic cell, because eukaryotes is distinguished in two ways. One is they have that is the distinguishment from the prokaryotes is in two things. That is, first is nucleus. So eukaryotes, they have a true nucleus, wherein their nucleus is covered by a nuclear membrane, whereas prokaryotes do not have a true nucleus, because the nucleus is not covered by a nuclear membrane. Eukaryotes, their organelles, whatever organelles are there in them, say for example the lysosomes, the mitochondria, the Golgi apparatus, the endoplasmic reticulum, all these are covered by membrane. It can be either a single membrane or a double layered membrane. But only ribosomes in eukaryotes is not covered by a membrane. Whereas in prokaryotes, none of the cell organelles are covered by, they have only very few cell organelles and which are not covered by any of the membranes. So that is one of the major difference. Now talking about the double membrane bound organelles, wherein Each of the cell organelle is covered by a double layered membrane. So which are those? They are the mitochondria, which is called as the powerhouse of the cell. Then comes the plastids, the Golgi apparatus, and the endoplasmic reticulum. So all these which I have written here, no, under double membrane bound organelles, mitochondria, plastids, Golgi apparatus, endoplasmic reticulum. So all these are double membrane bound organelles. So in plastids, again it is of different types based on the pigments that they carry, or based on whether they have pigment or whether they do not. So chloroplast contains carotene and some of the green pigments in them. Then chromoplast, then it has leucoplast, which are colorless plastids. Now these leucoplasts, so these two, that is chloroplast and chromoplast, they impart color to the plant. Okay, but leucoplasts, they are colorless plastids, and they do not impart any color to the plant, but they have another function of storage. That is, based on what material they store in them, it is divided into amyloplast, which stores starch, elaioplast, which stores fats or oils, and alluroplast, which stores proteins. Alluroplast usually found in monocot seeds, so it stores proteins. Next, talking about endoplasmic reticulum. Endoplasmic reticulum is again of two types depending on whether they are covered with ribosome or whether they are not covered by ribosome. So if the endoplasmic reticulum surface is filled with ribosomes, or if ribosomes are present on it, then it is called as rough endoplasmic reticulum, and if ribosomes are not there, then it is called as smooth endoplasmic reticulum. And endoplasmic reticulum is the only organelle which forms a continuity with that of the nuclear membrane, and Golgi apparatus is the one which is present somewhere close to the nucleus, but it has no any connectivity with that of the nuclear membrane. And endoplasmic reticulum and Golgi apparatus, they come under the endomembrane system itself because they depend on one another for their function. Next, talking about single membrane bound organelles, one of them is lysosomes, which are also called as the suicidal bags of the cell. And these lysosomes also come under the endomembrane system. Next, talking about the non-membrane bound organelles, as I told you, ribosomes. Ribosomes are the only cell organelles which are not bound by any membrane, be it in a prokaryote or in a eukaryote. Next, moving further, let's study about the nucleus. So nucleus, it contains the nuclear membrane. Then inside the nuclear membrane, it contains the nucleoplasm, which is a fluid matrix. In the nucleoplasm, chromatin fibers will be distributed, which is nothing but made up of chromosomes, and they contain the nucleolus. Now, depending on how the chromosomes are arranged, it is of different types. Like how the centromere in a chromosome is arranged, it is of different types. Metacentric means the centromere will be at the center. Submetacentric means slightly above the center. Acrocentric means even slightly above the submetacentric. Telocentric means almost at the tip of the chromosome. So that is why the two arms or sister chromatids will look very smaller when compared to the other two arms. That because of the position of the centromere there. So this was about the synopsis, like what and all is there under this particular chapter for you to recall. So next, let's move on to the 2-mark question and answers from this particular chapter. So briefly describe the cell theory. This is one of the important questions. Here, it was proposed by Schleiden and Skivan that you have to mention. And if you don't write the year also, it is okay. But mention the scientist's name who proposed this particular cell theory. And they made a statement. So you have to write the two statements. That is very important. So the statements that was given was all living things are made up of cells. So if you write the two statements also, it is enough. So all living things are made up of cells and products of cells. And one more statement that the cell theory gave was all cells arise from pre-existing cells. So these two statements, if you write, you will get 2 marks. Next question, list out functions of the cell wall. So you have to write 2 functions for this, okay, because it's 2 marks, 2 functions. So cell wall provides rigidity and shape to the cell, and cell wall forms an outer boundary and it is protective in function. So these 2 functions you write, you will get 2 marks. Next question, write the functions of plasma membrane. Again, you can write depending on, you can write 4 or you can write 2 depending on. Sometimes to avoid confusion, they will only ask write any 2 functions, write any 4 functions, it will be asked. So functions of plasma membrane, it allows passage of only certain selected materials. That is why plasma membranes are selectively permeable in nature. And it involves the process of osmosis and active transport. It helps in maintaining the shape and form of the cells in both plants and animals, because plasma membrane is present in both plants and animals. And it also helps in ingestion of liquids and solids. So these are the functions of the plasma membrane. Moving further, list out any 4 differences between plant cell and animal cell. Now again, the question is differences. So make 2 columns. On one end you write the plant cell and in the other column you write the animal cell. So first, talking about the cell wall, plant cell, cell wall is present. Animal cell wall is absent. Plant cell, chloroplast is present. Animal cell, chloroplast is absent. Plant cell, centriole is absent. Animal cell, centriole is present. In plant cell, they have a large central vacuole. Here, the vacuoles are quite small and many in number in animal cell. Next question. What are plastids? Mention their types. So, if you write what are plastids, you will get 1 mark. Mention their types. If you write, you will get 1 mark. So, what are plasmids? They are nothing but pigment containing cell organelles which are found in plant cells, that you have to mention found in plant cells, because you don't get to find plastids in animal cells. Plastids are the cell organelles present only in the plant cell. They are of 3 types: chloroplast, chromoplast, leucoplast. Moving further, list out the functions of Golgi complex or Golgi apparatus. Any 4 function you can write. The first function is modification and package of proteins. Then acrosome of sperm is produced by the Golgi complex. Golgi complex helps in the production of lysosomes, and Golgi complex help in the formation of glycolipids, glycoproteins and glycolipids. So any 2 if you write also, it is enough. So modification and package of proteins you can write, formation of glycoproteins and glycolipids also you can write. So next question. Mention the types of chromosomes based on the position of the centromere. So 4 types are there. Each will carry half a mark. So therefore 2 marks you will get. Metacentric, submetacentric, acrocentric, telocentric. These 4 you write, you will get 2 marks. Next question. Mention the different shape of cells with some examples. So different shape of cells that is there, that is the, say for example, the red blood cell will have a different shape, the squamous epithelial cell, nerve cell will have a different shape. So usually this question is rarely asked. So the cells differ from each other in shape and size. They may be disc-like, spherical, rectangular, cylindrical, polygonal, columnar, cuboidal, thread-like and elongated, or thread-like, flat or irregular. So for example, any one example if you give also, it is enough. I have given many examples here. Red blood cells, they are round and biconcave. White blood cells, they are usually amoeboid in shape. They don't have a particular shape. Columnar epithelial cells are pillar-like, or you can write they are finger-like. Squamous epithelial cells are flat. Nerve cells are branched and long. Tracheids, that is tracheids in the case of xylem, for example, they are elongated. So mention any shape of the cells with some examples. So 1 or 2 examples also you can write. So you write the shapes and write the example. Next question. Differentiate between gram-positive and gram-negative bacteria. Now make 2 columns. Gram-positive bacteria, they are stained by gram stain. That is why they are called as gram-positive. Gram-negative bacteria, they do not take up the stain or gram stain. That is why they are gram-negative bacteria. And gram-positive bacteria, they take up the stain because their cell wall is thin. That point you can write. Cell wall is thin. Here, gram-negative bacteria, they do not take up the stain because the cell wall is thick, and therefore the molecules that are present in the stain cannot pass through the cell wall. So, cell wall is thick. So, these 2 differences you write on each side, you will get 2 marks. Next question. Differentiate between cilia and flagella. This is also one of the important questions. Cilia, they are short hair-like and they are quite more in number. They are in lakhs together number. Flagella, they are long whip-like and they are very few in number. So for example, bacteria, they can have 2 to 4 flagellas, but cilia, they are hair-like, just like how a comb looks, no, or the bristles of a brush, how it looks, like that cilia looks. So 2 differences you write, you will get 2 marks here. So next question, what structural and functional characteristic of cilia, flagella and centrole have in common? Now here, we are not writing the differences. So we are finding out which structure and function in cilia, flagella and centrole is common, that you have to write. The common structural features of cilia, flagella and centrole is that they have basal bodies as starting point, and these basal bodies are arranged in 9+2 arrangement of triplets of microtubules. Functionally, they show contraction and expansion causing movement. So that is their function. So because of the contraction and expansion only, these organisms are capable of moving. So 2 functions, that is, you have to write about the structural similarity and their functional similarity. Or one point under each of that, you will get 2 marks. So next question, differentiate heterochromatin and euchromatin. So heterochromatin on one end, euchromatin on the other. Under heterochromatin, they are deeply stained or deeply stainable part of the chromatin. They are lightly stainable part of the chromatin. Heterochromatin replication is slower. Why? Because they are tightly packed. So that is why. Euchromatin replication is faster. Heterochromatin, they are less active. Euchromatin, they are more active. So because it is for 2 marks, any 2 points if you write on either side is also enough. Next question, why are lysosomes called suicidal bags? This is also one of the important questions. Lysosomes are sac-like structures which are bound by a single membrane. They are a single membrane bound organelle, no? And they contain some hydrolytic enzymes. And when these enzymes are released, when these hydrolytic enzymes are released, they cause breakdown of various cytoplasmic structures. And also, they are capable of engulfing the dead cells. So, that is why lysosomes are called as suicidal bags. Next, we'll move on to 5-mark questions and answers. So, 2 5 marks will come from this particular chapter. So, 10 marks, easily you can write. So, all the big question and answers, if you study from this chapter, no, 10 marks you will get, because compulsorily 2 will be asked in the exam. Any 2, 5 marks will be asked in the exam. And among those, this one is very important. With a neat label diagram, describe the fluid mosaic model or describe the lipid bilayer. Also, they can ask like that. So, you have to draw this diagram exactly from the NCERT textbook, wherein you'll have to mention about the sugar, the protein and the lipid bilayer that it contains, cholesterol and the here, this is the extrinsic protein, this is the integral protein. So much you have to mention. Whatever labeling is there in the NCERT textbook, that you have to mention. For this answer, you have to write, after drawing, you have to write the answer, and don't draw and leave it as such without labeling. Because without label, however beautiful and big you draw the diagram, it is of no use. It will be considered 0 itself. So please label the diagrams, and always don't draw a very small diagram. Let it be at least half a page. Next answer, that is explanation for this. S.T. Singer and G. Nicholson proposed the fluid mosaic model, and according to this model, it consists of a bilayer of phospholipids and proteins, and the polar hydrophilic head part of the lipids are located near to the surface, whereas also giant protein molecules are distributed randomly, and peripheral proteins are also present. They lie embedded or totally buried within the lipid bilayer, and apart from that, they contain few oligosaccharide molecules also, and the proteins and lipids are arranged in such a way that they exhibit semi-solid and semi-fluid property, or instead of writing semi-solid and semi-fluid property, no, you can write quasi-fluid property. Quasi-fluid means semi-solid and semi-fluid, and that is why proteins are capable of moving in whichever direction they want within the lipid bilayer. So that is why it is called as fluid mosaic model, which gives it, and this particular quasi-fluid nature gives it the flexibility. So much points you have to write. So next question, this is also very, very important. Usually it appears in the exam. Describe the structure of mitochondria with a neat label diagram and mention its functions. So you have to draw the diagram which is there in the NCERT textbook. You have to write about the outer membrane, mark the outer membrane, mark the inner membrane, the fluid matrix that is there. Then you have to mark the intermembrane space, and you have to mark the cristae, which are nothing but the inner membrane that folds, that is called as the cristae, which therefore increases the surface area of the matrix. So that is about the cristae. Don't get confused. Cristae, the foldings in the Golgi complex is called cisternae. The foldings in the mitochondria, we call it as the cristae. So let's describe the structure. So size and all that, if you don't write also, it's not a problem. First, we'll start from the membrane because it forms the covering, right? So mitochondria is a double membrane bound structure. Inner membrane has many folds, and that fold is called as cristae. And between the outer and the inner membrane, there is a space, and that is called as the perimitochondrial space. And on the inner surface of the inner membrane, there are knob-like particles called Racker's particles, and their main function is an electron transport system. I hope you remember this ETS, electron transport system. And the mitochondrial matrix it contains, inner to the, or the inside of the mitochondria is filled with the fluid matrix, which is called as mitochondrial matrix, and they're nothing but granular fluid substance. And in the mitochondrial matrix, it contains prokaryotic ribosomes will be present, DNA and several enzymes necessary for Krebs cycle will also be present. Next, moving on to functions. It is the site for aerobic respiration and also ATP generation, and hence it is called as the powerhouse of the cell. And mitochondria, they also provide different intermediates for the synthesis of cytochrome, anthocyanin, etc. So these are the two functions of the mitochondria. Moving further, next question, with a neat label diagram, describe the structure and function of chloroplast. Now this is also very, very important. Structure and function of chloroplast with the diagram. Usually it appears in the exam. So 3 questions are very, very important in this. One is the fluid mosaic model. Next one is the mitochondria, and the next one is the chloroplast. Out of these 3, any one usually appears for the exam. Next, for this chloroplast, they are the green plastids, that is we had studied, right? So they command the plastids and they impart green color to the plants. They are called as chloroplast, and they are present in the cytoplasm of all the green plants, and it is in the chloroplast where photosynthesis occurs because of the chlorophyll pigment. They are oval, spherical, even some of them can be ribbon-like, they can be reticulate also, and if you don't write the size also, it is fine. It is also covered by 2 membrane, and between that is the outer membrane and the inner membrane, and between the 2 membrane there is a space which is called as periplastidial space, and inside the inner membrane they contain a fluid substance which is called as stroma. The stroma it is colorless. It contains a lot of enzymes which are needed for photosynthesis. Apart from that, it contains circular DNA, RNA, and ribosomes are also present in them. So much you need to write about the chloroplast. Next, moving to, so in the stroma, what is there? So in the stroma, what is the function of the stroma? So say for example, stroma, it is the site for dark reaction, and in the stroma there are certain membrane bound flattened sac-like structures which are called as thylakoids, and each thylakoid are arranged one above the other like a plate, and it is called as granum. And the thylakoid are the ones which contain the photosynthetic pigments, that is chlorophyll, carotenoids, xanthophylls, whatever is there, which imparts different color to the leaf of the plant, that is present in the thylakoid. And grana, it is the site for light reaction. So light reaction occurs in the grana. This is also very important. Light reaction occurs in the grana, and the dark reaction occurs in the stroma. Okay. So dark reaction doesn't mean that it occurs in the dark. No, it is only thing is it occurs during the sun, when sunlight is there only, but it doesn't make use of the sunlight. That's it. Next, talking about the function, it is the site for photosynthesis. Photosynthesis is their main function and their only function. Next one, draw neat label diagram of plant cell. So plant cell, this also usually they ask either plant cell or animal cell, both of that also is very important. So this though it is quite easy for you because since your high school itself you have been drawing the plant cell and the animal cell. What are the main differences here? For plant cell, draw both the cell wall and the plasma membrane, but for the animal cell, draw only the plasma membrane because they do not have the cell wall. And in plant cell, one of the major things is they'll have a large central vacuole. Because they have a large central vacuole, the nucleus is always pushed towards the side, towards the periphery. Whereas in animal cells, they have vacuoles, but it is small and distributed, and the nucleus is at the center. That is one of the major difference. Apart from that, chloroplast you can mention here. Their animal cells do not have chloroplast, but these have chloroplast. Apart from that, all the other things are quite same, or the cell organelles are quite same in both the plant and the animal cell. So this is about the plant cell, this is about the animal cell. So all this you have to see, as I told you, the nucleus is almost at the center, and centrioles are present in animal cell, but you don't get to find them in the plant cell. So that is also one of the important things. Next, moving further. With neat label diagram, describe the structure and function of the nucleus. So, draw the diagram of the nucleus wherein you'll have to write about the nucleolus, the nuclear pore you need to mention, and wherever the nuclear membrane is not covered, no, it leaves a gap or a pore that is called as the nuclear pore, and it is that nuclear pore is important for the nuclear content, that is the chromosomes and all that to come out. It is covered by a nuclear membrane and it has nucleoplasm. So much markings that are there in the NCERT you have to mark. There are 4 components forming the nucleus. One is you have to write about the nuclear membrane. What is there in the nuclear membrane? It is double layered and it has many pores which are called as nuclear pores. Write about that. And these nuclear pores connect the nucleoplasm with that of the cytoplasm, so that the exchange of the contents between the nucleoplasm and the cytoplasm can occur. And the space between the two membranes is called as perinuclear space, that is the outer membrane and the inner membrane of the nucleus. Next, we'll write about the nucleoplasm, what is there in that. It is also called karyolymph or karyoplasm because most of the essential components of the nucleus is present there, and it has dense fluid filled protein granules. It has some free RNA and some enzymes. Next, it contains chromatin fibers or chromatin network, and it contains chromatin threads, and the threads later on during the process of cell division they condense, and that process is very, very important for the mitosis and the meiosis. Next, it contains nucleolus, which is spherical structure, and it usually disappears. It is there. Nucleolus is seen only during the resting stage of the cell, that is in the interphase. Later on, when they get into the division process, right from the prophase until the cytokinesis, they will disappear during the cell division. And apart from that, in the nucleolus, protein and RNA are also present. Next, talking about the function of the nucleus, it is the main component of the cell which helps in controlling all the activities of the cell organelles. And it controls all the metabolic activities of the cell, and it is a place for, or it is a reserve for the genetic material, and it helps in the synthesis of the ribosomes, and also the synthesis of RNA takes place in the nucleus. So these are the functions of the nucleus. So next question, this is also one of the very, very important questions. So all these which I put here now for 5 marks, all those questions if you study properly, easily 10 marks you will get, because 2 5 marks will be asked from this particular chapter. Next, this question, with neat label diagram, explain the different types of chromosomes based on position of centromere. So you have to write about draw the diagrams, that is you have to draw metacentric, then you have to draw the submetacentric, then you have to draw the acrocentric, and you have to draw the telocentric. So these images you have to draw, and how the labeling is given in the NCERT textbook, same way you have to mention about the satellite. You have to mention about the secondary constriction, then you have to mention about the secondary constriction, then you have to mention about the centromere, the short arm, then you have to mention the long arm also. So much you have to mention. So you have to explain about this. That is how the position of the centromeres are. Based on that, submetacentric, acrocentric, telocentric and metacentric chromosomes you have to write. It is easy because if you know the image, then you can write the answer looking at the image itself. So for example, metacentric means exactly at the center. So the centromere will be present exactly at the center. So therefore, both the arms of the chromosomes or the 4 sister chromatids that are there, they are of equal size. Then submetacentric means it is slightly above the center. Therefore, 2 arms will appear a bit shorter when compared to the other 2 arms. Acrocentric means slightly above the submetacentric. Therefore, there will be 2 shorter arms and 2 longer arms. Telocentric means it is almost at the tip. So therefore, 2 arms are very, very short and 2 chromatids are very longer. So much these points, if you write under each of these, it is enough along with the diagram. So this was about this session. So whatever 5 marks I gave from this, any 2 will surely come for the exam. So study that very well. So this was about the session. I hope you found the session useful. So we shall meet again in the coming session discussing the important questions under a new chapter in exam point of view. So see you in the next session. Thank you.